Hello everyone, my name is Veronica Go, and I'm currently a senior at the University of Tennessee in Knoxville, Tennessee, and I'm a double major in biochemistry and statistics. And today I want to talk to you about the effects of peripheral beta blockers on isoprenaline-induced fear condition rats. So, what exactly is anxiety? Well, anxiety can actually be defined as a psychological disorder that causes feelings of nervousness, worrying, and fear, and it actually affects approximately 18% of the US population. You might be wondering, if it causes nervousness and fear, why would you even want anxiety? Well, evolutionarily speaking, anxiety was used to detect potential threats, such as a lion charging right towards you. Obviously, you want to be able to react to a lion that's charging right towards you. So the anxiety would be able to activate the fight or flight response. Hence, you would be able to fight off the lion or run away from it. However, in today's society, anxiety is largely socially constructed, such as when you're taking a test or perhaps giving a big presentation to your boss. That causes anxiety, but in today's society, anxiety is rather maladaptive. Biologically speaking, the beta adrenergic system is the responsible factor for symptoms of anxiety, such as increased heart rate, sweating, shortness of breath, shaking and trembling, weakness, and the list goes on and on. So, as you may or may not know, the picture that is depicted below is actually the beta adrenergic system. Epinephrine binds to the receptor. The receptor activates the G-coupled protein. The alpha subunit dissociates from the beta and gamma and goes to the adenylate cyclase, which then allows for ATP to be turned into cAMP, which activates the pKa, which is responsible for many different pathways such as fat breakdown for more energy, sugar breakdown for more energy, etc., etc. Within the beta adrenergic system, there's actually two different types of beta adrenergic receptors, the beta 1 receptor and the beta 2 receptor, and each is responsible for its own task. So the beta 1 receptor is responsible for the heart. It increases the heart rate, increases impulse conduction, increases contraction, and increases ejection fraction. On the other hand, the beta 2 receptor is responsible for blood vessels, such as dilating small coronary arteries and dilating arterioles. There's also two different types of drugs that can affect each of these receptors, agonist and antagonist. An agonist is something that will affect the system and actually allow it to activate. An antagonist does exactly the opposite. It'll bind, however, it will have no activation of the system. But if you put an agonist and an antagonist together, you're going to get an attenuated effect in the sense that you will have less activation of the system. So this is basically what my research was trying to do, trying to find an agonist which would allow for the induced anxiety, but also trying to find an antagonist that would reduce the amount of anxiety. Using pharmacological techniques, I was able to induce anxiety as well as reduce anxiety. So the first drug that is on this list is isoprenaline, which is actually a synthetic analog of adrenaline. It's a non-selective beta adrenergic agonist, which means that it will affect both beta 1 and beta 2 receptors, and the fact that it's an agonist means that it will activate the system. The next drug is nadolol, which is a peripheral non-selective beta adrenergic antagonist. It's peripheral because it only works in the peripheral nervous system. It's non-selective, meaning, once again, that it'll work on beta 1 and beta 2 receptors, and the fact that it's an antagonist means that it will theoretically reduce the effect of the agonist, which in this case is isoprenaline. Then we have atenolol, which is a peripheral selective beta 1 antagonist, meaning that it'll only affect beta 1 receptors and theoretically affect the agonist effect in beta 1 receptors. There's actually two different doses of atenolol because I wanted to see if it was a dose-dependent effect. Finally, I used saline as my vehicle and my control group. In order to test for anxiety, both immediate as well as learned, I used an olfactory fear conditioning paradigm, which basically takes the Pavlovian classical conditioning technique, which pairs a neutral stimulus, in this case amyl acetate, which is an odor that smells vaguely like bananas, with an unconditioned stimulus, which is isoprenaline, which would normally affect the rat and induce anxiety anyways. I chose to use the olfactory fear conditioning paradigm because olfaction is actually important for the rat's survival, such as choosing the best food to eat or choosing the best mate to reproduce with, etc. In addition, olfactory cues are associated with emotional learning, and this is associated with neural networks within the brain that have to do with olfaction as well as emotional learning. For my experiments, I followed a 4-day protocol which consisted of familiarization, conditioning, conditioned stimulus 1, and conditioned stimulus 2. On day 1, I let the rat explore the cage with no odor and no injections to make sure that they're not fearful of the cage initially. On day 2, I pair the drug with amyl acetate, such as the agonist or the antagonist, to condition the rat to be fearful of the amyl acetate. On the 3rd day, I actually switch boxes into a box that has a enclosed space, which is right here, as well as an open space, and the amyl acetate is placed in a tube that is usually in the corner. The different box is actually used in order to ensure that the rat does not feel a fear because of the cage, but rather because it's worried about the odor. And the enclosed space is actually helpful for the rat because it feels safer. And so if it experiences fear towards the odor, it'll run towards the hidden space rather than approaching the space with the odor in it. And on the 4th day, I use only the box with no injections and no odor to see whether or not it created a fear association with the box itself and where the amyl acetate originally was in the previous day. So I actually did 2 different experiments. In experiment 1, I tested for saline versus isoprenaline, saline being the control. And this experiment was basically done in order to see whether or not isoprenaline would be an effective way to induce anxiety within these rats. In experiment 2, I wanted to see if I could use different drugs to reduce the isoprenaline-induced anxiety. So I injected saline plus isoprenaline, nadolol plus isoprenaline, or atenolol plus isoprenaline, and saline, nadolol, or atenolol was injected 15 minutes before isoprenaline. And like I said before, it was to see whether or not I could reduce the amount of anxiety felt from the isoprenaline. In addition, using a video tracking software called Animaze, I used the software to track behaviors such as defensive behaviors like freezing, which is when the rat is fully immobile, and crouch sniffing, which is when the rat is immobile but the nose is moving and sniffing the air around it. I also tracked the exploration time, which is the amount of time the rat would explore the cage freely, and I also accounted for the hide time as well as the approach time, which hide time is when the rat is in the enclosed space of the odor box, and the approach time is the amount of time it spent near the amyl acetate or where the tube was originally placed the day before. These are the results I got from experiment 1 and experiment 2. In this chart, you can see the defensive behavior that was tracked in both experiment 1 and experiment 2, and this is all put together. As you can see, the familiarization session does not have any difference between the groups, but the training, which is just the conditioning days, do have differences in the saline group and the both atenolol groups, they clearly display differences as opposed to everything else. However, the exploration time did not display any type of difference between the familiarization and the training groups. When I looked at the approach time as well as the hide time, which the approach time is when they approached the amyl acetate odor, and the hide time is when they spent the time in the enclosed space, I actually calculated the percentage of time that was spent in those zones over the overall time. As you can see in the percent approach chart, everything besides saline and nadolol was different from the groups. On the other hand, the high time was all similar except for the 20 milligram per kilogram dosage. Finally, I tested for test defensive behavior as well as test crossing. Test crossing being how many times the rat would cross between the enclosed space and the open space. And this was done for both condition stimulus 1 and condition stimulus 2. As you can see, the test crossing did not display any differences between any of the groups. However, in the amount of tense defensive behavior, which is crouch sniffing and freezing, both atenolol groups and saline plus isoprenaline group was displaying much more defensive behavior than the saline, isoprenaline, and nadolol plus isoprenaline group. So what do my results actually tell me? Well, if you look at the charts, you can actually see that atenolol did not seem to reduce fear condition anxiety. The most obvious way to tell whether or not it actually reduced anxiety would be to look at the amount of defensive behaviors displayed, and the amount of time that the defensive behaviors are displayed as well. Nadolol, on the other hand, seemed to reduce fear condition anxiety and actually attenuated the fear acquisition, meaning that they didn't feel fear during the conditioning session, and it attenuated the amount of consolidation, meaning that they felt less fear during the CS1 test, which is the conditioned stimulus 1 test, as well as the conditioned stimulus 2 test. This suggests that the peripheral nervous system does have a role in producing anxiety-like effects, and that if we target beta receptors non-selectively in the peripheral nervous system, we can allow for the reduction of overall anxiety. These are just some of the references that I used, and there's definitely way more references that you can refer to in my paper, which is also on this website. And finally, I would just like to acknowledge the MERT program, Minority Health and Health Disparities International Research Training at Christian Brothers University, which is in Memphis, Tennessee. And I would like to also thank the Padua Lab, which is pictured below, at Universidad Federal de Santa Catarina, which is in Florianopolis, Brazil. Thank you for listening.